So this provides an overview of the clinical studies score, which is one of the components of RADx Tech. And a lot of our work over the last 3-4 years has led to updated guidance from FDA for rapid antigen testing, specifically surrounding serial testing. Uh, some of the research studies we have done has contributed to a test agnostic machine learning based reader, as well as identifying testing anomalies. We continue to, as of last year, validate the performance of antigen tests with new variants, as well as validating digital biomarkers for the viral prodrome. On the left slide of the screen shows how we started and pivoted from summer to winter of 2020. So early on, some of the legacy studies that we had under RADx Tech clinical studies course were called COVID-19 Test Us. And it was a platform design where point of care technologies, like you can see on the two pictures on the left side of the screen, were being used at testing sites across the country at 10 different locations and compared against gold standard PCR testing. But with the introduction of rapid antigen testing and the need to generate more home-based data collection, we switched to a decentralized study approach and adopted a no data left behind strategy where we were able to collect user-generated images, wearable device data, preference for reporting antigen test results to public health, symptom diaries that complemented standardized symptom checklist, and are planning on launching a sub-study later this year that would also look for acoustic biomarkers related to viral prodrome. On the right side are some of the screenshots of impactful findings and dissemination, guidance and amendments from FDA for use of rapid antigen testing, a schema that highlights the ways we were able to use patient participant generated images to create automatic classifier of test results, and a New England Journal of Medicine article where we were able to report this rare but now known phenomenon of certain people, especially those with autoimmune conditions, always testing positive with certain rapid antigen tests. And so this slide provides an overview of the studies that we conducted under the clinical study score. COVID-19 Test Us and Test Us Kids were device validation studies. They were single time point tests by design. Investigational device were compared against Roche Cobas PCR assays. All of the testing happened in person, and the data were generated with the primary purpose of supporting an EUA application. Because of the nature of this design, the company's identities are masked for data that's made available through Data Hub, and instead they correspond to a device ID that were assigned to each of these investigational devices. COVID-19 Testers Bank is the third row here, and something that I would come back to at the end of these slides, but it's a biorepository study where we collected various sources of biospecimen from symptomatic and asymptomatic participants that tested positive as well as tested negative. And this is a study where we collected samples from across the country, as well as some samples from South Africa. Home antigen testing studies, both to understand feasibility and efficacy, started in winter of 2020. These were longitudinal studies to understand how well and for how long people can do rapid antigen testing. That informed some of the more real-world evidence generating studies like Stop COVID-19, where we were able to provide tests to participants at their home in a very decentralized manner, and then ask them to take an antigen test and confirmatory PCR test when they were symptomatic. We also asked participants to share their wearable device data and take images of each antigen test that they took. Test Us at Home has actually two different flavors of the study. This was the serial asymptomatic testing study done in collaboration with FDA, where we enrolled asymptomatic participants who had not previously tested positive for COVID-19 in the previous few weeks. We started with doing paired 48 hours testing where every 48 hours for 15 days, they took rapid antigen test and PCR collection from home and shipped the PCR samples or the nasal swab samples for PCR assays to a central lab. All of the participants took images of each antigen test, and we were able to sequence a fair number of the PCR's collection swabs. We switched the 48-hour cadence to 24-hour for a subset of 600 out of the 7,200 participants to understand more granularly the testing performance in a longitudinal way. And then COVID Detect was another study where we wanted to understand viral dynamics after exposure, and those were similar to Test Us at Home, longitudinal studies done post-exposure or post-positive test, but they're along with antigen test that was point of care as opposed to at-home antigen test. We also did a PCR assay and a viral culture. So this provides an overview of where we are with data upload. Most of the survey data, tier 1 and tier 2 and 3 have been uploaded and are available in Data Hub. Images from home studies and Stop COVID-19 are cleaned and harmonized. We have cropped them to make sure that identifying information or non-image or non-test images are not made available through the Data Hub, and will be making it available later this year. And we have wearable device ready and will be likely collaborating with RADx DHT to ensure harmonization and standards across the different RADx subgroups. And sequencing data are available for Testis Bank as well as some of the other studies. And so I'll end on just a announcement that we are open for requests for the biorepository. We have nasal swabs, saliva swabs, blood samples, dried blood spots, as well as plasma and PBMCs.
To find, access, and begin analyzing studies from this and other RADx data programs, navigate to the NIH RADx Data Hub at radxdatahub.nih.gov. Navigate to the Study Explorer, where you can use filters or text-based search to find studies of interest. The RADx Data Program filter allows you to filter studies by RADx Data Program. Once you find a study or studies of interest, click the study name to navigate to the study overview page. Then click the dbGaP study accession link to navigate to dbGaP to submit a data access request. It is important to note that the email you use for dbGaP must match the email you use for your RADx Data Hub account. Once you receive an email notifying you that data access has been granted, log in to the RADx Data Hub and navigate to the My Approved Data tab underneath Data Access. To learn more about how to use the RADx Data Hub, visit our helpful information page on the website, view our other videos, including our Getting Started playlist, or contact us at radx-datahub@nih.gov.